Okay, my name is Reese,
My name is Emma.
and we're going to be testing how temperature affects the rate of growth of Tetrahymena. And we hypothesize that if you place it in an environment with a temperature of 30 degrees Celsius, then the rate of growth will be slower than that with an environment with a temperature of 37 degrees Celsius.
Our dependent variables are the length of time that the Tetrahymena cultures are in the specific water baths, which is 24 hours, and also the amount of the Tetrahymena cultures that we will be using. Our independent variable is the temperatures, which will be at 37 degrees Celsius and 30 degrees Celsius. Our control group will be the Tetrahymena that are not subjected to the different temperature. And that's so that's 37 degrees Celsius. And then our experimental group will be the Tetrahymena cultures that are subjected to the lower water temperature at 30 degrees Celsius. We've randomized this treatment by selecting the Tetrahymena cultures and making them into two separate groups to determine their growth while subjected to the different temperatures. And we will have 2 replicates per each water bath for the 37 degrees Celsius water bath and the 30 degrees Celsius water bath.
Just a brief overview of how we're going to set up our experiment. We're going to have our initial amount of Tetrahymena in a 20 milliliter beaker, and we're going to use sterile pipettes to transfer 5 milliliters into 4 10 milliliter test tubes. Then we're going to take initial cell counts of each test tube. We'll then put them in their specific water bath treatment with 2 replicates per treatment. And after the 24 hours is over, we're going to retake a cell count to get our final cell count. And we're going to make a graph using the time and the cell count as our X and Y axes. For one of our methods that we're going to use, we will be using, we're going to measure the amount of Tetrahymena. We're going to measure the growth of the cultures after 24 hours in the constant temperatures to see if there was a growth or a decay of the Tetrahymena.
We predict that the Tetrahymena cultures will grow the most abundantly in the water bath that has the temperature of 37 degrees Celsius because it is the common temperature that the cultures typically live in. We believe that the temperature will allow the natural growth of the cells rather than the colder temperature.
Yes, some of the materials, or I suppose all of the materials we will be using are water baths. We're going to have 2 water baths, as we've said. It'll be 30 degrees Celsius and 37 degrees Celsius. And then we'll need 20 milliliters of Tetrahymena stock cultures. We'll need 4 10 milliliter test tubes, a beaker, which is 50 milliliters, and pipettes to put them in their test tubes. And that's it. Thank you.